Welcome back, everyone. Today I have a quick lecture on water and electrolytes and acid and base balance. Just a brief outline for today, just one slide introduction. We'll talk a little bit about water and electrolytes, where they are. A little bit of it is a review of active and passive transport. We'll talk about the plasma and interstitial fluid and how molecules can move around there. The movement of these electrolytes, and talk about the kidneys, and then acid-base balance. I think we all know that water and electrolytes are essential for physiological processes, but these aren't static. There's a constant exchange with the environment, and the content is dependent on how much we take in and how much we lose. The exchange typically takes place at two barriers: transfer through the cell membrane and then at the capillary walls. And we'll talk more about these. And the kidneys play a major role in the regulation of the water and electrolyte homeostasis. Let's first talk about water. Water is obviously essential for survival. About 60% of the adult body weight is water. In newborns, that percentage is higher, and it's lower in older adults. About 60 to 90% of cellular composition or weight is water. In the brain tissue, that number is really high. It's about 90%, whereas in fat tissue, it's pretty low, around 10%. Water in the body is either in the intracellular fluid, that's about 2/3 total of the water in our body. This intracellular fluid is the fluid within cell walls and the cytoplasmic fluid. And then the extracellular fluid is about the rest of the water. This is the interstitial fluid, which would be just the fluid between our cells, the fluid in the lymph system, the fluid in plasma, the fluid in transcellular fluids such as gastrointestinal fluids, urine, and cerebrospinal fluid. The capillary wall separates plasma from the interstitial fluid. So shown here, it would be a cell with the cell membrane, the interstitial fluid shown here. The capillary wall is freely permeable to water and electrolytes, but not proteins, as we've talked about in the past. So you can imagine a capillary moving through the interstitial fluid here. You can get passing of, for example, sodium and potassium ions, but not necessarily proteins, shown here. You can also have passing of these electrolytes across the cell membrane, as we talked about in the past. Let's talk a little bit more about electrolytes and what is meant by the anion gap, and talk a little bit more about plasma versus interstitial fluid. The body fluids differ in ionic composition. So here's a chart. I don't expect you to memorize this chart, but it's a useful resource for you. But you can see things like plasma versus gastric juice versus small intestinal fluid. These have different levels of electrolytes such as sodium, potassium, bicarbonate, and chloride. For example, there's a higher level of bicarbonate in plasma. It acts as a buffer. You can see the differences between bile and sweat, for example. The majority of electrolytes are present in plasma and the intracellular fluid. So, for example, you can see in plasma there's a lot of sodium, a lot of chloride, bicarbonate, as I mentioned. And the concentrations of ions and low molecular weight molecules are similar in plasma compared to the extracellular fluid. You can see the differences between plasma and intracellular fluid. Intracellular fluid has much higher potassium. In the plasma, there's total concentrations of around 150 millar sodium in plasma. Again, don't worry too much terribly about the numbers here, just trying to give you a sense of the electrolytes in the different fluids, whereas potassium is much lower. So, the point is not that you know the exact concentrations, but high levels of sodium in plasma relative to potassium could come up on the exam, for example. The most abundant, so those are the cations you can see here. The most abundant anions are chloride and bicarbonate. So the rest of the anions constitute what's known as the anion gap. So if you look at the concentrations that we showed on the previous slide, if you add up all the cations and subtract the anions, so the phosphate, sulfate, protein, and organic anions, for example, lactate, citrate, pyruvate, acetoacetate, some of those other organic anions. Anion accumulation is clinically important during renal failure and diabetic ketoacidosis. The electrolytes in the intracellular fluid are shown here. The main cation is potassium, so there's the difference between the intracellular fluid and the plasma. That's much, much greater, 30-fold greater. The main anions are proteins and phosphate in the intracellular fluid. Okay, let's talk about the movement of electrolytes. We've talked a little bit about this in the past. We'll go a little bit more into it. What the sodium and potassium pumps, passive electrolyte movement, and some of the clinical conditions associated with the movement of electrolytes. As I mentioned, water diffuses freely across most cell membranes, but the movement of ions and neutral molecules is restricted and/or highly regulated. So small molecules are transported by transport pumps, as we've talked about in the past. So for example, the sodium-potassium pump, it maintains the sodium and potassium gradients across the cell membrane. Here's a diagram of the sodium-potassium pump. It's an ATPase that drives movement of sodium and potassium across the cell membrane. It can be considered an ion transporter because it's a sodium pump, but it's also an enzyme because of this. It is an ATPase. It hydrolyzes ATP. It's electrogenic. It remains an electrochemical gradient across the membrane. It hydrolyzes one ATP molecule, and the energy release drives the efflux of 3 sodium ions from the cytoplasm to the extracellular space, and in turn the influx of 2 potassium ions from the extracellular space into the cytoplasm. The movement of water is linked to the sodium transport, so this pump is critical to water absorption in the intestine and reabsorption in the kidneys. So the pathophysiology of hypertension and chronic diarrhea are linked to impairment of this sodium pump in the kidney as well as the small intestine. We've talked a little bit about passive electrolyte movement in the past. This can occur through channels. It can be driven by energy of electrochemical gradient, so such as sodium, glucose, amino acids, phosphates. We talked a little bit about this in the past. When we have movement of water between the intracellular fluid and the extracellular fluid, it can cause differences in osmolality. So for example, when you have normal osmolality, you have the same concentrations of water on both sides in the intracellular and the extracellular fluid. The change in concentration of ions that can decrease or increase the osmolality of either the intracellular or extracellular fluid in either one of these compartments, it creates a gradient of osmotic pressure, and consequently water can move either in or out. So if you have decreased ECF, extracellular fluid osmolality, water is moving from the ECF to the intracellular fluid. until a new equilibrium is reached. And then in the opposite direction, if you have increased ECF osmolality, water moves from the intracellular fluid to the extracellular fluid. It reaches this new equilibrium. In that case, when water is moving out, it can lead to cell dehydration. You don't have enough water in your cells. Whereas if water is moving in, it can lead to cellular edema. Basically, your cells have too much water. Normally, water always diffuses from lower osmolality to higher to equalize osmotic pressure. Sodium is abundant in the extracellular fluid, as we've talked about, and thus is important to determining osmolality. Glucose is normally low in the plasma with no effects on osmolality, but during diabetes it can become a major determinant as well. When we talk about clinical abnormalities, they may develop after fluid loss, depending on the composition of what's lost. For example, sweat contains less sodium than the extracellular fluid, so therefore excess sweating leads to hypernatremia, which means your serum sodium concentrations are pretty high, greater than 145 millimolar. A predominant loss of water basically concentrates sodium in the extracellular fluid or in the blood. On the other hand, the sodium content of the intestinal fluid, is similar to that the sodium content in the plasma, but it contains considerable amounts of potassium. So severe diarrhea would result in dehydration and low blood potassium levels. This can lead to significant decrease in plasma, but may not change sodium concentrations. The last few slides here, I'm going to focus on the kidneys and acid-base balance. The kidneys have an obvious important role in the acid-base balance and water osmolality. It maintains the composition, osmolality, and volume of the extracellular fluid and controls this acid-base balance. The glomerulus is a cluster of nerve endings, spores, and small blood vessels in a particular cluster of capillaries around the end of a kidney tubule. You can see the glomerulus here. We get an exchange of water, sodium and potassium and hydrogen ion exchanges in the kidney. The glomerulus, as I mentioned, the plasma filtration occurs in this region of the kidney. So its composition, osmolality of the filtrate changes as it flows through renal tubules. About 80% of the filtrate is reabsorbed, and the sodium potassium ATPase activity is a few thousand times higher in the kidney to make sure that we get reabsorption of sodium. Here's just a cross section of the glomerulus as shown here. We'll finish here with acid-base balance. So as we talked about, the normal physiological processes result in the generation of a variety of acids, such as carbonic acid, uric acid, lactic acid. For example, carbonic acid is generated as carbon dioxide in tissues, and can dissolve water. This results in the release of hydrogen ions. The blood hydrogen ion and pH remains constant. However, the hydrogen ion is around 40 nanomoles, and the pH is around physiological pH around 7.4. If we get changes in pH, it can affect the ionization of protein molecules, and obviously then this could lead to impacts on enzymatic activities. A decrease in pH would increase sympathetic tone and may lead to cardiac dysrhythmias. So to orient you here, if we're talking about blood, tissues, kidney, and the lungs, the blood and the tissues contain buffer systems that minimize changes in hydrogen ion concentration. So the lungs control the exchange of CO2 and O2 with the blood and the external environment. The red blood cells transport gases between the lungs and the tissues, and the kidneys control plasma bicarbonate synthesis and excretion of H plus, or hydrogen ions. The main buffering systems in the human body, I mentioned bicarbonate being one of them in the extracellular fluid. Phosphate buffers are big in the intracellular regions. Proteins are also buffers in the intracellular regions, and hemoglobin is a good buffer in red blood cells. In saliva, bicarbonate buffering systems are very important, as shown in the extracellular fluid on the chart here. As I mentioned, intracellular buffers are primarily proteins and phosphates. The hydrogen ion from the plasma goes into cells in exchange for potassium moving out. So an accumulation of hydrogen ion in the plasma, so you have, in that case, you have acidosis, low pH, acid concentrations, acidic concentrations in your plasma. This leads to hydrogen ions moving in and potassium moving out, so you have higher plasma levels, or higher potassium levels in your plasma. And on the other flip side, you can have alkalosis, where you have a deficit of hydrogen ion in the plasma, so your pH is higher, and this can lead to low plasma potassium concentrations because you're moving hydrogen out from the cell and then potassium in. As I mentioned a couple times, though, the bicarbonate buffer system is essential for maintaining the acid-base balance and regulating carbon dioxide waste. This allows for maintenance of relatively constant plasma pH, and it counteracts any force that would alter plasma pH just through this simple buffering system, having carbon dioxide and water to make bicarbonate, and then this dissociates into the hydrogen ion and the bicarbonate ion. We talked about the Henderson-Hasselbalch equation in the past, so we can just think about this for the bicarbonate system. pH is equal to the pKa plus the log of the anion over the acid. You also have to use a solubility coefficient because carbon dioxide is not fully soluble here. So the pH in the plasma is driven mainly by bicarbonate, and you can derive it using this simple equation here, where the concentration of bicarbonate is around 24 and the concentration of carbon dioxide is around 1.2. Again, this minimizes changes in the hydrogen ion concentration and therefore stabilizes the pH when you either have acid or base added to the blood. When you have acid added, it reacts with bicarbonate and it forms carbonic acid, which dissociates to form carbon dioxide and water. So this helps neutralizes the excess acid that may be added to the blood. On the flip side, when alkali is added, it reacts with carbonic acid to yield water and bicarbonate. So this does cause bicarbonate to increase slightly. And then what happens, you get, because of this increase in bicarbonate, you get subsequent decrease in carbonic acid. So the excess OH is basically neutralized in the end, and the depletion of CO2 is compensated by a decrease in ventilation. Let's dive into this a little bit deeper, the components of this bicarbonate buffer and how the blood and the kidneys and the lungs interact here to maintain this pH. So the blood pH is proportional to the ratio of the plasma bicarbonate to the partial pressure of the carbon dioxide in the blood, as we showed with the Henderson-Hasselbalch equation there. The components of the bicarbonate buffer are carbon dioxide and bicarbonate. The partial pressure of CO2 is called the respiratory component of the acid-base balance, and the bicarbonate is called the metabolic component. So the metabolic component is more relevant to the kidneys, whereas the lungs, when we have things in the gaseous state, this would be the respiratory component. Both the lungs, which is the respiratory component, and the kidney, the metabolic component, contribute to maintaining the pH in the blood. The two components are closely related. When we have a primary disorder that is respiratory, it causes accumulation of carbon dioxide, and thus you get a compensatory increase in the bicarbonate reabsorption by the kidneys. So the kidneys absorb more bicarbonate in this case. Conversely, when you get a decrease in the partial pressure of CO2, it causes decreased bicarbonate reabsorption. When the primary problem is metabolic, you get a decrease in bicarbonate concentration, and thus resulting in a decrease in the pH. This stimulates the respiratory center to increase the ventilation rate. So it blows off more CO2, and then the plasma partial CO2 concentration decreases. So this is why you get patients with metabolic acidosis, you get hyperventilation in these patients. On the other hand, an increase in plasma bicarbonate concentration, which causes an increase in the pH, leads to a decrease in the ventilation rate and CO2 retention. This change always helps return the pH to normal. So that was a lot of words, but it basically boils down to this simple diagram here. So if we think about the blood pH as a concentration of the bicarbonate anion to the partial pressure, the concentration of CO2, if we have acidosis conditions on the left and alkalosis conditions on the right, you can essentially see that it boils down to this simple equation. So if we have metabolic acidosis, that's because the bicarbonate concentration is low, so the numerator is low here, leading to a lower pH. Conversely, if you have respiratory acidosis, it's because your CO2 concentration is high in the lungs, the high number on the denominator here, again, lowering pH. But in both cases, you have a process that results in a decrease of the blood pH, because you have higher concentration of the hydrogen ion. The opposite is true for the alkalosis. So in this case, metabolic alkalosis is you have high bicarbonate concentrations in the kidneys or low CO2 concentrations in the case of respiratory alkalosis. And again, the opposite effect is here. So higher number in the numerator or lower number in the denominator raises the pH level. And so you have in either case, you get an increase in the blood pH, essentially a lower concentration of hydrogen ion. So that sums up this lecture. Here's the study guide for today. Here's some take-home messages that I want you to get from this lecture. Essentially, where the water is found throughout the body, I mentioned at a high level, 2/3 is here, 1/3 is here. That kind of information is useful. Where the electrolytes are found in the body, again, I'm not expecting you to know the exact concentrations in the plasma, for example, but it's good to know relative concentrations. For example, sodium is high in the plasma versus other other fluids. The main cations and anions in the body, I went over those. The what is the anion gap and how is it filled? So that is an important thing for the exam. I had a couple slides on the sodium potassium pump. So which ions move in and out is important, and the fact that it is not only a pump, it's also an enzyme that uses ATP hydrolysis to drive that movement. The clinical conditions associated with water and ion imbalance is important to know. What is the role of the glomerula and as part of the kidney? And then the last slide, really, the last couple slides, I talked about some of the main buffers in the body being proteins, hemoglobin, but really I focus mainly on bicarbonate and the CO2 system. And so really that last slide that I just shows understanding how that works and how you get an imbalance there in your blood pH and how the different systems of the kidney, the lungs, and the blood are really connected through this buffering system.